Pessoal, vamos começar? Hoje, como a gente tem uma palestrante internacional, eu vou fazer uma breve introdução em inglês, tá? So let's start. All right, let's begin our conference of our lecture series. Today we have the privilege to welcome a leading expert in the field of immunology and neurobehavioral disorders, Dr. Judith Van de Water from the University of California, Davis. Briefly speaking, she's had a long and impactful academic career making groundbreaking discoveries about how the maternal immune system can influence neurological development, especially through her work on maternal autoantibodies as a risk factor for autism spectrum disorders. With over 190 scientific publications and prestigious international recognition, Dr. Judith is a global reference in her field, and we are truly excited to have her with us today. Thank you very much, Judith, and the floor is yours.
Thank you so much. Usually only my mother calls me Judith, but so Judy. So thanks, everybody, for coming today. And for anybody who is at the meeting in Fortaleza, I apologize. There will be some overlap. But I think just as an introduction to where I come from, as part of the UC Davis M.I.N.D Institute, and the institute was built by parents and of children with autism to try to better understand the medical side or what we could potentially do for autism. So as part of the institute, there's a number of different disciplines. I'm used to speaking to very multidisciplinary audiences because there are so many of us that work together from different fields, all sort of trying to understand the underpinnings of autism. And I will try to not speak at really fast speed. So I think, one of the questions we ask a lot, I'm not a clinician, I'm a Ph.D. immunologist, but I still work in a very translational, so always working in the clinical population as well as the animal models. But I think one of the questions we ask is, what if we could predict which individuals may be affected by autism? And I think one of the things that we know is the earlier we can detect, the better we can do interventions and potential therapeutics. So this is always our goal. And I think just to understand autism for anyone who doesn't work in this field, I'm sure everyone knows someone who's impacted by autism, but I think the core features to remember, it's a behaviorally defined disorder. So we have very few biomarkers, very few molecular markers. It's really diagnosed based on the child's behavior. And the other aspect of that is it's diagnosed usually between you can start seeing signs at 2, but more likely 4 or 5, which is late in development a little bit in children because they have some neuroplasticity still, but it's a little bit late. We don't understand the causes, but what we do understand is that there are likely during gestation, maternal factors that may be influencing how the brain develops, and those maternal factors are in the work I do or in the immune system. So the maternal inflammatory environment has been described as, I think, first really described in schizophrenia. I think that they were a little bit ahead of us on that in understanding that the birth season may impact how the brain develops in the offspring. And so I think there's some overlap with schizophrenia in that area. And then just as background, the autoantibodies that recognize self-proteins have also been described as changing the way the fetus develops. And this was really, the biggest precedent for this is systemic lupus, SLE, where children from mothers who have autoantibodies or have SLE often are born with congenital heart blocks. So that maternal autoimmune disease is impacting how the heart develops. So I'm going to talk a little bit about gestational immune dysregulation. And when I say that, I think we probably would interpret that as inflammation. But I hate to use that word because sometimes it's not pure inflammation. It's just changes in the immune profile of the mother. And there's a lot of epidemiologic evidence that suggests changes in the maternal immune system, how that impacts the development of the brain in the offspring. We see this, we are now looking at, as I said, in autism. The earlier work was largely around schizophrenia. So when we think about the immune response, I think the important thing, and I've always worked in autoimmunity and clinical immunology, and the one thing that we really think about is that balance between mounting an effective immune response, because that is really important, and sort of keeping that immune response balanced. I think we saw this really during COVID with the lung, sort of the impact of COVID on lung, that sort of, the word cytokine storm, which we've always used, became like a popular word in the mainstream, but that cytokine storm suddenly became known to people. But I think it's this balance and regulation is what we look at as to why some people, when they get sick during pregnancy, might go on to have a child with a neurodevelopmental disorder, but not everyone does, right? So there's some underlying, likely genetic reasons for that. So I think one of the areas that we're very conscious of is that the immune signaling molecules play a really important role in how the brain develops. And I think also an important thing to remember, we call them immune molecules because that's where they were first described, but lots of cells make them. Lots of cells respond to them that are not immune cells. So when they're in the central nervous system, they likely are, we wouldn't think them as immune, but rather growth factors, signaling factors, factors that tell cells where to go, chemotactic factors. But the nervous system and the immune system co-develop, and it's a very important sort of partnership during neurodevelopment of those two systems. Let's see. So as I said, the majority of women exposed to immune challenges during pregnancy give birth to perfectly neurotypical children, right? It's not just if you get sick, you're going to have a child with autism. And I think probably the similar thing to Zika virus, right? Not everyone who's exposed to Zika virus has a child with microcephaly, but it is how you're responding to it, the level of response. Those are all the things that we look at, sort of, have you met sort of a threshold of responsiveness? So I think, as I said, genetic susceptibility, the genetic underpinnings of the mother, and the intensity of that immune response, and how sustained that immune response was. How long does that go? We have I'm not talking about today, but because this is all clinical data, but we have a non-human primate model of maternal immune activation, and how long the dam responds, the mother monkey responds, is really important into what the offspring behavior and what their immune system looks like. So there's definitely a relationship there. So what do we know from our various human studies? I think I do a lot of both human clinical and then animal studies. And one of the first studies that we worked on was the early markers of autism, or EMA study. And this is, we were looking at autism and developmental delay risk factors in a clinical population. So this was a Southern California population of women. Samples were taken, oops, I know I have a picture of it. They were taken during that early second trimester period. And these were taken and banked for another reason, but we had access to those through a colleague of mine, and were able to measure the cytokine chemokine profiles. And one of the things that we did was compared mothers of children with autism, without autism. But I think the highlights that I'm going to show are mothers of children with autism with no intellectual disability, and mothers of children with autism with intellectual disability was the big defining, and then developmental delay without autism were the defining populations. And just looking at the cytokine chemokine profile, the ASD versus GP, there were some differences, but the biggest differences were with ID against general population controls. When I say general population controls, these are the children had not been seen to see where they had developmental delay, autism or not. They were just sort of a random sample of the general population. But the biggest difference was we were defining in these various cytokines and chemokines, we were seeing the elevation really distinguished the population that had autism with intellectual disability from autism without intellectual disability. So this is a sub phenotype, to use the word that we use, of this population was based on how high the levels of inflammation were in the mother during this second trimester period. And we have just, different elevations in different innate Th1, Th2, Th17 cytokines, and innate chemokines. So I think this was an example of starting to look at what might be driving some of the differences we see within autism. Autism is a very broad spectrum. You have from very high functioning, very brilliant, we think of the prodigies, down to very low functioning, no language, severe intellectual disability, and that is the population that we're distinguishing with this profile. Oops. So what we saw, as I said, the mothers of children with ASD with intellectual disability had inflammatory cytokines and chemokines, and that it separated them from developmental delay without autism. So those look quite, they both have intellectual disability, but the etiology seems to be quite different between those two populations. So to us, it suggested that there was potentially a lack of immune regulation in this population. So I'm going to switch topics a little bit to what is probably the biggest part of my work, which is maternal autoantibodies in autism spectrum disorder. And this, for us, was a fairly new paradigm that we developed in my lab. And just because I'm an immunologist, because I worked in autoimmunity, I think we think everything is caused by the immune system. But we started to look to think about, because we knew of other autoimmune pathways to as I said, like SLA, changing how the fetus develops. And so we started thinking about that in terms of autism. So this is an area that we actually came up with a name for, maternal autoantibody-related autism. And this is where some women, so IgG crosses the placenta around day 100 in humans, day 12 in rodents, to protect the fetus from everything the mother's been exposed to, right? So it's a passive way of protecting the fetus because the immune system hasn't developed yet. And so unfortunately, if autoantibodies also cross the placenta, they will cross with the healthy IgG and could have a negative impact on the fetus. And one of the things, and I'll show you some detail in a minute, we saw definitely that these antibodies were getting into radioglial stem cells very early, and that these antibodies were changing the way the neurons are developing. This is present in about, it depends on the population. It's interesting, we've seen some very big differences in sort of location of where the women are living, sort of their demographic profile. So it's anywhere from 12% to 26% or 25% of women who have a child with autism have these autoantibodies. So it's not all of autism. Nothing is all of autism. But that is sort of what we've seen. And we've noticed that this particular subtype of autism is a little more severe, and also depends on the pattern, and I'll explain what I mean by that in a second. So they have worse stereotypic behavior, lower language, lower function, and in some cases, more intellectual disability. Unlike sort of, I think an important distinction is the maternal immune activation model, I think, which is people are more familiar with, where we use a viral mimic to create sort of a sickness in the mouse models or rat models or monkey models. This is different from that, and it has a very different profile as well. So we identified from a very crude Western blot is how we started to realize that there was potentially something there that was very specific. And then we went on to determine what the proteins are. So STIP1, stress-induced phosphoprotein 1, cyprinter-GDA guanine deaminase, neuron-specific enolase, lactate dehydrogenase, which is in every single cell. That one is a little bit surprising. YBOX1, which is a transcription regulator, and then CRIMP1 and CRIMP2, which I think to me are the most interesting. Those are a little bit later. So each of these has a different role in how the neurons develop. But the latter two, the CRIMP1 and CRIMP2, are really part of that maturation process of the neuron. And you'll see later that they have, that combination has the biggest impact. And I think the thing to remember, you never see, just antibodies to just one of these does not give you pathology. But it takes a combination of two or more to get you above the threshold of having a negative outcome. And so many of the women that we see have more than one. So they'll have, say, CRIMP1 and CRIMP2 and NSE and STIP1. And those children have very severe autism. So if you have we have a scale that we use 1 to 10, or 1 is the least severe and 10 is the most severe on the ADOS severity scale. Those kids are at the 10. So these are very highly impacted. So that effect is additive. As you keep adding antibodies into the profile, it has a worse impact. So we wanted to create, because we want this, actually, this is part of translating this back out to the public, is to create a test that we can use to predict if a child will have autism or not earlier than the behavior assays can do. And so we created an ELISA. It took 4 statisticians to do this project, to understand what, we used some machine learning to understand what the combinations were that were specific for autism, the antibody combinations, and then which ones were clinically relevant. So when we were doing this, this is postnatal samples. So these were taken postnatal, the child, we had the mother's blood taken at the time the child was diagnosed. But we still saw this very high specificity. So an odds ratio of 31 is very good, right? That's a very high risk, 31 times more likely. And this is with 100% specificity. When we say precision here, that is what we mean, that they're 100% specific for autism. We don't see them in any other disorder or in controls. And so these, when I say we have combinations of 2 or more, that's what I'm talking about, with the top 2 being CRIMP1 and GDA, and CRIMP1 and CRIMP2, and NSE and STIP1. So that was our top clinical patterns. We also noticed that if a woman had antibodies to CRIMP1, she had the severity of her child's autism on that ADOS severity score went up to at least 2-fold. So with CRIMP1 as the part of the pattern, you got a more severe autism. And we're seeing that in our animal models as well. So that was the postnatal sample. In the prenatal samples, same sample I just talked about, the cytokine and chemokine responses, we did the same. We looked at the antibodies in this population. So now this is taken during gestation. Thank goodness, when we did this, that the patterns we described in the first studies held up in this. So we tested those patterns in a different study population. The interesting thing is, oops, there we go. So there are different, this was a different, geographically different study population. And the most abundant pattern here was CRIMP1 and CRIMP2, where CRIMP1 and GDA was way down at the bottom. So that was one interesting thing, is where the samples arise, which probably speaks a little bit to the etiology about how they are breaking tolerance in the first place. But we saw the same thing, very high specificity. But I think more interesting is that we saw differences in the patterns with respect to intellectual disability. And a couple of these patterns were very highly specific to having a child with autism with intellectual disability, where a couple of the other patterns, which we call, they're a little less abundant, were more in the no intellectual disability phenotype. So for us, clinically, this can be important for when we're giving this information to providers, because it helps them understand what the phenotype of this child will be, right? How severe their autism will be. So for this, we found reactivity to any of the patterns was highly accurate in their ability to predict risk. Reactivity to CRIMP1 and CRIMP2 was associated with a more severe autism with intellectual disability. And some patterns would distinguish, as I said, the ASD with and without intellectual disability. So I think this facilitates access to early intervention care. And as I also said, we see, in some cases, we don't see very high CRIMP1, CRIMP2. We see higher NSE STIP1 in some populations. So it's kind of interesting. So I think we know that these antibodies are very good biomarkers, and that's what we're looking at them for right now, right? How well do they predict? But I think for us, a very important question is, are they pathologically significant? Meaning, if they're present, are they doing damage? Are they doing anything? And so we first used passive transfer models to determine the effect on outcome. And we've done a couple of non-human primate studies at University of California. We have a primate center, so we're lucky to have the ability to do that. But what we noticed is when we inject the dam with human antibody, so this passive transfer of pathologic antibodies, we saw one interesting finding was the dams were overprotective. They didn't let their offspring leave them much, which is really bad for socialization and social education of the offspring. As they got older, they showed evidence of abnormal social behavior, not that they avoided it, but whatever behavior they were projecting, the control stimulation animals that we were testing them with was not liking that behavior. So they would not sort of, they call it affiliative behavior. That was sort of their deficit. So species typical affiliative behavior, meaning whatever they're communicating is not well received. And we also, so you can see just by the graphic that the controls here, very normal, typical species, social development, where our treated animals were very flat. They did not really develop social skills, and I will show you. I don't know how well you're going to be able to hear it, but you can hear. So these are two controls where we have a stimulus animal and one that was treated with control antibody. And you see very normal behavior. They're interacting with each other. So this is just post-weaning. The animals are placed in this sort of a play cage. It's got a lot of toys and things for them to climb on. There's a little mounting behavior. So normal 6-month-old juvenile monkey behavior. And this will take a minute. But they're very cute. They cuddle. They're very, very social animals. One goes to sleep, so they're clearly not stressed. And these are animals they were raised with. So an untreated animal, so they know them. They've known them their whole life. Obviously, they're buddies. Sorry, this will take a second to get to the... So one of the stereotypic behaviors, you can tell which one came from the treated, potentially. So this is one stereotypic behavior, which is pacing. And that animal, the minute it goes in there, all it does is pace. And what you'll notice is, the other animal goes nowhere near it, because this is very weird behavior, and they are not going to interact with those animals. So that is one of the stereotypic behaviors. And I think the next one, I forget how long this movie is. Oops, oh shoot, that's not what I meant to do. Yeah. And here's the other behavior. And you can hear how loud they are compared to the other ones. This one has the back flipping stereotypic behavior, and all it wants to do is just backflip. And again, the other one is like, oh my God, get me out of here. I do not want to be in here with this crazy monkey. It's like, please. So you can see that was our first sort of indication that we had that these antibodies were doing something pathologic. When you do autoantibody research, you always first test passive transfer because it's very easy to do. Harder to do is breaking tolerance. But so we also noticed that we saw larger brains, that these animals were developing, their brain was developing differently. And this is a theme that you're going to see recurring. So then we wanted to, so this is our, these are our monkeys. And you can see the difference here. And these are our humans. So these are brain scans from humans. And we looked to see which had just autism without the autoantibody, which had autism with the antibody, and then the typically developing controls. And you can see that the MAR-ASD brain, and these are in boys, are larger than both groups, so significantly larger. So there's a physiologic effect of these antibodies being there during development. So the other passive transfer experiment we did, this is a very artificial exposure because we're literally, my colleague Veronica Martinez-Odeño at UC Davis is a neuroanatomist, and they're very good at injecting into the ventricles of the developing fetus. This is on day 14 of these embryos, and then on day 2 hours later. So we took IgG from patients, and we labeled it with biotin, and then we injected it. So they're biotin-related antibodies, and then these were the MAR antibodies. These are controls. And literally, it was so clean, I thought we hadn't labeled it, that we'd screwed up. But it turns out we did label it, because we did it again. And we see these are injected here into the ventricle, and this is the subventricular zone, the ventricular zone and the subventricular zone, and these are radioglial stem cells. And you can see these antibodies are actually getting into those cells. And this, when you work in autoantibodies, I have colleagues who've been in this area for a long time of autoimmunity, and they're like, no, it has to be extracellular for the antibody to see it. And I think we've shown, she argues with me every time she sees this, and I said, no, they're there. They are inside the cell. And I think there's a permissiveness of these developing radioglial stem cells that allows the antibodies to get inside. But instead, I think the other sort of misconception is if you have an autoantibody, that you're killing cells, that you're destroying tissue, but actually, it's just changing the way these cells are developing, and it accelerated their migration from the ventricular zone through the subventricular zone into the cortex and caused the increased mitosis. So it is triggering, these are again the same antibodies with the LDH, AB, CRIMP1, STIP1. So it is changing how those cells are developing. So you can see we have way more cells in this population in the subventricular zone. So the other thing that we saw in this is that depending, and these are 3 different brain regions that Veronica looked, and we saw using Golgi stain a big difference in the dendrite. So we saw dendritic spines were almost absent or blunted. You'd see some, but there was definitely an impact, and I think one of the potential issues with this might be because there's that normal migration process is very important for those radioglial stem cells to be educated and develop properly, and it's like we're just shooting right through it, right? So they're not getting the normal time to development that they should have. So passive transfer models are great, but they are not as good as what we call a clinically relevant endogenous model, or where we make the animals make the same autoantibodies that we see clinically. So we mapped on we epitope mapped, so we mapped on these proteins where the clinical antibodies were binding. Then we took those peptides so that the animal, our mice or our rats would make the exact same antibody profile. And then we did behavior. We looked at the brains in the rats, and in the mice, we did MRI, and we did our MRS spectroscopy, and a number of studies. So one thing, we've done both C57 Black 6 mice, that's what the first one we did, and the second was the rat, and we got the same outcome, which is also good. So it doesn't matter, the genetic background does not matter if these antibodies are present in the animals. And so the mouse data are on the top, but I think it's even better to watch sort of the video. Excuse me, the video. And so you'll see we have a control animal and a stimulus mouse. So the dot is on the saline exposed animal. The stimulus animal actually is worse behavior than our control animal, but there's a lot of interaction. They spend time in the cage together, and sort of the metrics for all that are in the graphs on the top. But what we did notice is that, and you'll see in a minute, the MAR-ASD mouse, you'll see it's pretty easy to tell which one it is, and the stimulus animal keeps trying to engage, and all that the MAR-ASD animal wants to do is self-stim, so it's self-grooming, and you can see it just goes to the other side of the cage. It does not want to interact. And the poor little stimulus animal keeps trying and it keeps getting rejected. Because mice are not quite as smart as rats, we moved to rats because they have better behaviors, more intricate behavior. But you can see there's just an avoidance, a social avoidance. And the 3-chamber task did not work as well for this model as the juvenile social dyad did in separating these animals out. The other thing that we noticed, and this is most often we do USV calls in the very early pups. I have rat data for that. But what we also tried is we put adult males that were MAR-exposed males in with an estrus female, a female that in estrus, which should excite them greatly. And if they're a control animal, it does. These are all the voice print calls. You can see right when they're put in the cage, these are taken at the same time. So these prints that I'm showing are from the same time frame. You can see the males are very excited and they're very vocal and they're interacting because in the MAR-ASD, there's nothing. I mean, it's just like they just don't even care that there's a female in estrus in the cage with them. So that was another sort of it's like a social interaction, a little bit verbal, but we can't say that mice are verbal. And one of the things that we did, if you're familiar with what clarity imaging, where you remove everything but the structural proteins, so you remove all the lipids, and then you can image the entire, so this is an entire, from those mice that I showed you the behavior, postnatal day 2 brain. And they were exposed, so these were exposed to LDH, CRIMP1, CRIMP2, or sorry, LDH, A and B, CRIMP1, and STIP1 during gestation. So the animals that the dams we made the antibodies in. And you can see, and this is actually one of those happy accidents because my graduate student had ordered antibodies. Anti-IgG was supposed to be for staining other cell structure antibodies. And he goes, the antibody control is staining positive. And he realized he ordered mouse anti-mouse. So it's a happy accident. So when he did it, what he saw was IgG. This is just anti-mouse IgG. So all we're staining is the IgG that had infiltrated into the brain. So this is after perfusion. So the animals were perfused before the brains and washing. And you can see on the left is the MAR animal, and this is control. I'm going to say the postdoc that perfused did not do a great job because there's a lot of IgG in the vasculature there, and you can see it. But we did see that the IgG was bound to the target proteins, and I think if you can see on the screen, it's very intracellular because 2 of these targets are quite intracellular. And we saw, so you can see some mitotic cells as well. So that just was further proof to us in our active model. And so we moved to rats, partly, as I said, because they have better, more sort of a higher level of social interaction and social play. And so we did the same paradigm as we did in the mice. And in the rats are a little bit different. But you'll see how much, so these are the stimulus animal and the control. And the stimulus animal is in red and the control is in the black. And you can see they play a lot as juveniles. I mean, they roll around. They're like the monkeys. They have a great time together. They actually like this task. And if you can pick which one is the MAR animal, 2 interesting things about this. The MAR is the one with the dark on it, and you can see it is not really engaging. It's looking around the cage. But the other thing is that the control rat is feeding off the behavior of the MAR rat and not really trying, and it just stops and just licks the cage for the rest of the time, right? So that's its stereotypic behavior. But unlike the mouse that keeps trying, the rats are they're smart, right? And they're like, this behavior is not normal. I just need stay away from this animal. Oops. So we also did some neuroimaging in adolescents at postnatal day 30 and postnatal day 70. And we noticed there was definitely a change in striatum motor cortex and the somatosensory cortex and corpus callosum, which were larger in the MAR offspring. So there were, again, like the humans, like the monkeys, and like the mice, because we also did it in mice, we see this elevated brain volume. But the other thing that we noticed, so we saw a decrease in communication, decrease in social behaviors. You saw an increase in grooming, which rats don't do. Rats don't do repetitive behaviors like mice. Mice, it's very common. And I remember our students that were doing all the behaviors, like, you got a really bad batch of rats. They're really weird. And they had no idea what the experiment was. And it turns out that they were just having this elevated grooming. But we also saw just an increase in the brain volume, but we also saw, we saw altered brain metabolite levels, which is a different way of looking at what is going on in the brain. And one of them, which I can't read from here, there we go, the decreased glutathione, cysteine to taurine, that pyruvate is necessary, and LDH is necessary. That enzyme is necessary for shuttling between pyruvate dehydrogenase, which I worked on in a former life, and then lactate dehydrogenase. So there's potentially, we're seeing some ramifications of those antibodies being present. But this is a nice way of understanding sort of just metabolically what is going on in the brain. So then we went on to remember the clinical patterns I showed you of CRIMP1, CRIMP2, CRIMP1, GDA, and NSE and STIP1. All the models I showed you to now was sort of from our early work, so we had defined that pattern. So then we went on to create what we call our more clinical patterns. So we went on to look at animals that were CRIMP1, CRIMP2, STIP1, NSE, and CRIMP1, GDA. And then we kept the controls in there. And then we looked at, we did behavior, which I don't have the data for yet. Behavior takes a really long time to code all those videos. But we also were able to collect brains from postnatal day 2, from embryonic postnatal day 2, 45 and 70 offspring. And we did RNA sequencing, and then we looked at cytokines and chemokines in the brain, not because this is a maternal immune activation model, which we would likely see changes in the offspring brain, but because we wanted to understand if there were changes in the growth factors in the brain of these animals, because my graduate student is very clever and thought that would be a good thing to do. So this is just showing that the antibodies are made in the dams where we don't see it in the controls, just to show the difference. But what we saw was not an increase in cytokines and chemokines, but we saw a decrease. So they're actually lacking in what should be present in normal brain at postnatal day 2, which is an interesting finding. So these are very important for healthy brain development, and it is looking like they are lacking in that. And then we saw several differentially expressed genes, with CRIMP1, CRIMP2 being the most different from controls within up and down regulated genes, followed by the pattern, our original pattern, CRIMP1, GDA being the lowest, and then STIP1 and NSE. And those genes are really what we determined was the enrichment. So the upregulated genes were all related to cell matrix adhesion and nervous system development, and the downregulated genes were related to synapse transmission and neuron differentiation, which I would expect given everything we've seen with how the cells are behaving and with the cytokine chemokine profiles that we're seeing. So really what we're seeing is just a lack of the normal pathways that we're supposed to see. So I think one of the, just in addition to behavior, as I mentioned, we have humans on the top, rats, mice, and monkeys all have these changes in brain volume. Males and females in the animal models, in the mice and the rats have it differentially. The volume is higher in some females and lower in brains depending on the brain region, but the behavior is the same. There's no sex effect. This is an equal opportunity way to get to autism, where we see normally is like 4-to-1 male to female ratio. It doesn't matter for this there's no sex effect of these antibodies. So as I said at the beginning, what if we could predict, and I think we are, which children would be affected by autism? I think the MAR autoantibodies suggest a potential biomarker of both risk and etiologic pathway in a subpopulation of women. So remember, not all of autism, but a very defined subpopulation has these autoantibodies. So just, I think, we discussed that immune dysregulation may contribute to risk of altered neurodevelopment. We know that the antibody is localized to the brain. I didn't even show all those pictures, but that both mice and rat models capture all three domains, the social communication and the stereotypic repetitive behaviors in a rodent, and that we see the enlarged brains in humans, mice, monkeys, rats. So to me, that piece that we just see, regardless of what species we're in, tells me these antibodies are definitely changing the way neurons are developing, and that, stay tuned for what that means, and that gestational exposure alters brain cytokine and chemokine levels. This is very new data, that they have differentially expressed genes, and I think that CRIMP2 says we saw CRIMP1 and CRIMP2 was the strongest, where these differences in cell matrix adhesion being upregulated. I don't know what that means, and I think it'll be interesting as we explore that. And then downregulated genes relating to synapse transmission and neuron differentiation. I think this is probably the biggest finding for this, and hopefully will give us downstream targets that we can, or maybe small molecule targets that we can potentially develop therapeutics. I have a great team of people I work with. There's actually probably more people that I work with. Knowing that I'm an immunologist, I have zero background in neuroscience, so I'm very dependent on students that come into my lab that are neuroscience graduate group and some of my colleagues. But most of the work that you saw today was done by Matt. Kat has taken over. Jana's about to finish. And she had a baby in the meantime, so she was very productive in lab. But I have some great students that have done this work, and I am happy to take any questions.
Questions?
So thank you very much for the talk. Wonderful results. So you described around 6 or 8 autoantibodies, right? 8. Mm-hmm. 8, yeah. So do they get activated during an infection? Well, what I want to ask is, are they always there? And if the mother has them, the risk is the same? Or if the mother gets a higher immune activation during pregnancy, do these antibodies go up and the risk is elevated?
So, and they're very consistent. Once the mother breaks tolerance, and we don't know how, as a matter of fact, Pedro and I are going to try to work on some things to understand that. But once the antibodies are there, they're there. And so every pregnancy thereafter is the same outcome. So there's not, from what I have seen so far, during pregnancy, there's not like a spike. Mind you, during pregnancy, IgG is elevated, right? Because you're producing more to passively transfer the fetus. But we have not seen a difference with pregnancy. I have a couple of cases I know of women who'd never been pregnant that were positive, and definitely one of them had very severe inflammatory bowel disease. So potentially other autoimmune sequelae. Infection, we're looking at that. Are there any infections that may have cross-reactive antigens? So I don't know the etiology, but I know the antibodies are pretty steady once they're produced. They don't change.
Wonderful talk. Thank you very much. I'm not familiar with autism. I work with development of the nervous system. So maybe you mentioned, I have two questions. One is about the brain size. Okay, both humans and the animal models have bigger brains, higher volume of the callosum, and when you show the radial glia, that is actually the neural stem cell, at that stage, I think is giving origin to neurons. So my question is this big volume due to more neurons or more astrocytes, for instance?
So we have not yet seen any impact of this on glial cells. But what we did, so Veronica, in the animals that she let develop to adulthood that we injected in, she saw larger cell bodies and more spaces between them, which is, I have no idea what that means, right? But so I think we're seeing a slight increase in cells, but definitely more space between the developing neurons, or the mature neurons, when we finally see that. So that is potentially what.
I mean, but it's very easy to count number of neurons. Anyway, this is.
So my graduate student Kat is doing that now, in the rats.
Yeah, because the larger volume of the callosum, since callosum are axons, so probably means that you do have more neurons, and that's kind of counterintuitive. That this results in more severe cognitive defects.
I think unless those neurons are incorrectly developed, right? So if those neurons have a lack of dendritic spines, they're not going to be communicating. They're just sitting there taking up space, right? So they are incorrectly
that's
functioning.
The second question.
Yeah.
You show less spines, there's dendritic spines that are actually the connections. But that usually happens after birth. I mean,
those were adult.
Okay.
Yeah.
Okay. So means that the antibody from the mother goes to the fetus and it stays there during the whole development, doing the trick. So the other question is, did you check the changes in the fetus? Could be something that the fetus read from the mother, and then his immune system or its immune system, because it has to be a continuous effect. Neuron generation, synaptic pruning, connections correctly, everything is postnatal. It has to be something very permanent. And so I was thinking if you can look the other side around, from the fetus side.
We are. So we have those brains, right? So we have the postnatal day, embryonic day 14 and 17. So we are doing the same study we did in the postnatal day 2 in those, because I'm very interested to see, especially in the RNA-seq, what is up and down regulated in those very early. Is it the same? We do know that the cytokine chemokine changes are early and are the same, and it lasts into early adult. So those growth factors present in the brain are not correct for quite a long time, right? That seems to be a consistent finding. We don't see the antibodies in the offspring once. In humans, it's about 9 months where the IgG from your mother is totally gone. In the rodents, it's shorter than that. It's like 3 months. But so they are still present even in the postnatal brain. So they could sort of be impacting some of that process. I think one of the things my student is doing is to understand all the changes that we're talking about, how, because we can do it in a culture dish, how they're impacting, what processes they are actually physically impacting of the developing neuron.
Hi, thank you so much. It's so puzzling, the presentation. Just wondering a little bit about, you didn't mention anything about the isotypes of the IgG, both in the mothers and also in the models. Because, I mean, in general, we would expect to have maybe subsets that are complement fixing. I'm not a neurobiologist, but I just came from a neuroimmunology meeting that we had in the Northeast, and I was very impressed about pruning. And there is so much data about, complement fixation. So I was just wondering, what do you know about this possibility that you have effector stage? Of course, I saw the data, and it is impressive. The antibodies, apparently, they are accumulating inside the neurons, right? So this was one side, and maybe the second has to do maybe with what she asked before. Network theory, it's out of fashion. We sort of got distant
But we
from
remember
that.
it, yes.
But in that particular case, maybe it would be worth revisiting, because after all, you could have anti-idiotypic antibodies maybe in the repertoire to sustain, some sort of cross-reactive activity with the tissue. So these are the issues that came to mind.
Yeah, and I think, so far we haven't seen complement with these. We see no damage. You see no
damage, no inflammation. There is no recruitment. So I mean, there is
it's very
evidence.
yeah. So, I mean, that's the first thing you look for, right, with autoantibodies. Is there some? But what we do need to look at is microglia and the pruning. That's the part that Kat is looking at now. Two things. How do they get into those cells?
Yeah.
Because they're definitely getting inside. So what is facilitating that? And then is there an impact on axonal pruning? Right? Is there something related to that? And complement certainly plays a part, right, in axonal pruning.
Hi, Judith.
Hi.
Nice talk. Thank you so much. Actually, I have two questions. I don't know if you have time. The first one is about the gut-brain axis. So both in patients and mice models as TLR agonist administration during the pregnancy, we have gut physiology alterations associated to autism symptoms. So in your model of autoantibody transfer, do you have microbiota or
We have not seen, we don't see a difference there. One thing I do see when we did PET imaging is there's a lot of autoantibody in the liver of the fetus. So there's possibility, not in the gut necessarily, but in the liver, that sort of that part of the gut-brain axis that we might be impacting.
Because dysbiosis is associated to worse symptoms, so could be. And my second question is a little bit about this, because I am wondering about the children born during COVID-19 pandemic. Those babies, most of them were exposed both to infection and inflammatory storm and or autoantibody, because we have an increase of auto-diseases induced by COVID-19. So do you have data about this?
We do, and that paper is back in my hands to resubmit after review. So when I get home, I have to resubmit that paper. But yes, we do see an increase in autism with COVID-19 infection, I think, as you would expect, but not as strong as I think people anticipated, which also speaks to how severe it might have been. We don't have the data on that, right? I mean, you have to hit that threshold to get that sort of really severe COVID infection. And so I think it'll be interesting. I mean, I do have the data for it. I don't have it memorized. My slide is not here. But we do see an impact, which is a very different model than this, for sure. I know we've got MIA models that, when I showed the metabolic profile in the brain, completely opposite from MAR. So the cytokine, chemokine profiles, very opposite. So those are really, really different. So that acute inflammation is very different than this chronic antibody present.
So the titers of your antibody are not different in the mothers that
So the titers of the autoantibodies?
Yeah, the titers of the specificity that you have described.
Yes, the titers are, I mean, some women have higher titers, but when we created the assay, we created it to have a threshold So we wanted to exclude the low antibodies because the specificity really was related. Specificity for autism was related to the threshold that you hit. So these are all very high, sort of in the clinical. The mice, we saw a little bit of differences, but not a lot because we immunized them with Freund's adjuvant and really to break tolerance to get a higher titer.
We have a question from our online audience. Adriana Bonomo is asking, what is the frequency of ASD in women producing these autoantibodies?
It's between 12 and 26%, depending on the population.
Really high.
Yeah. Yes. Yes.
Sure. Yeah, but the online audience,
they can't hear.
we need the mic.
Thank you very much.
Thank you.
Yeah, just a quick question, because you're talking about like in pregnancy, you have this increase of IgG. Everybody knows it's fine. But have you checked in your assay if you have a more prevalence of IgM positive mothers before the pregnancy or something like that? If you have any kind of correlation of high levels of natural IgM against your antigens in this case?
We did in the first early studies that we did, and we didn't see a big, that there was like a big level of IgM, then isotype switch. But part of the issue is how far back do you have to go to see that elevated IgM before you see the elevated IgG. You might have to do a very different kind of study than we've done. And that's a very expensive study to do. Pick people way before they even have a pregnancy, which is a great study to do, but can't afford it.
Any more questions?
Oh,
Whoa. Do we have time?
I'm not going anywhere.
Okay, just a little.
Until we're done.
Hello, Dr. David.
Hi.
Dr. Judy, thank you for the lecture. It was really grateful being here to listen to you today. So I also work on the autism spectrum disorder. I work with the model of operate, exposure during pregnancy. Yes. And I've seen during my studies a lot of correlations and a very high auto-risk ratio for siblings that have been previously diagnosed with autism disorder and maternal age during the pregnancy. And during the lecture, I was thinking that maybe this woman that had these higher levels of IgG could be keeping them during the development, and somehow this could be being a second factor for the development of ASD in their further children and the children that come afterwards. Is it possible to test it on your animals too, keeping these rats or these mice that have been? You did?
Yes. No, you're right, exactly. And once they're there, every subsequent child, which is about why we have that 25%, 26%, because those are often the high-risk moms that already had a child with autism. And we've seen if they were there the first time, they were there the second time. And then our rats, we do keep them, and we have multiple litters. And then, like the third litter, we do the embryonic, right? So and it's very consistent. A little bit different than the maternal immune activation model, which is not quite so consistent. I think it's a little harder model to work with.
That's great. Thank you.
Thank you.
I had a second question, if I may. So you have a very well characterized model of autism, right? And you also said that in other slightly different models that you saw, sometimes the metabolic pattern was inverted.
Oh, the maternal immune activation.
Yeah, so you have in your model, you are able to replicate those three pillars, right? Repetitive behavior, social communication. So what is, in your opinion, is the similarity between all of these models that give exactly this? You even show the rat stopping to vocalize, right? So what do you think is common to all of these situations? Do you think there is something common?
Are you talking about between maternal immune activation and this model?
And other models that
And other models of
we
outbreak?
have no idea what. So that give exactly the same.
I think So we have mice and monkeys that are maternal immune activation model as well. So I've got these, both things are going on at the same time. What we have noticed is, I think maternal immune activation is a very good model for altered neurodevelopment because of immune activation in the mother. I think one of the things that we've noticed is I think, and my colleagues and I think, it's a little better model for schizophrenia than it is for autism. And part of that is because of the delayed onset of symptoms in the offspring. And you especially see this in the non-human primate. You can really tell there's a little bit of a delay and more anxiety in that model than we see, certainly in this one. We don't have any anxiety in the maternal autoantibody model. It's a very different than some of the maternal immune activation. So that's one difference that we see, is that inflammation, I think, increases the anxiety behavior. Definitely changes eye tracking, but some of the symptoms, especially in the monkeys, as I said, are a little more delayed. So it's not that very early. We're seeing differences very young in our rats, for sure. We tried to make a maternal AMAR model in the monkey. It is very hard to break tolerance in a non-human primate because we can't use the same tools that we use in mice and rats, and sustain it because they have a very long gestation. So you have to, when you're breaking tolerance, so they resolve these antibodies. You can break tolerance, but they don't sustain. So that's something we've learned from the non-human primate. There has to be that activation for this sustained loss of tolerance to these proteins. You need a really strong hit to the immune system to break tolerance, to make those and make them last. So that has been a challenge in that model, where poly-IC is a pretty easy model to do in the monkeys.
Any more questions? Then let's wrap up our conference.
Thank you so much.
Thank you very much.
I love the questions.